Jonathan, could you hit the record button? Thank you. Thanks for joining, everyone. Welcome to this month's ECSA Files webinar. And we're really pleased to have Helmut Hillebrand today. Helmut is studied biology at the University of Oldenburg in Germany. And then he did his PhD at the University of Kiel. After a couple of years in Sweden, he returned to Germany, first to Cologne, but then he moved back to Oldenburg as professor for plankton ecology in 2008. And since 2017, he's the director of the Helmholtz Institute for Functional Marine Biodiversity at the University of Oldenburg. So we're really excited to have Helmut here today. And I'm very much looking forward to hear more about thresholds for biodiversity change and whether there are any. Thanks very much, Helmut. The floor is yours.
Thank you, Stephanie, and thanks everyone for joining and having me, and more than happy to convey a few thoughts that recently emerged out of discussions about how to best envision biodiversity change and management measures to mitigate biodiversity change. You see only my name on the first page, but of course the research I'm presenting is done by fantastic colleagues and PhD students. And instead of having a long pile of names on this slide, I will actually introduce them properly when I present their research. So biodiversity change has been noted as one of the two major environmental crises related but not identical to the climate change crisis. It has reached dramatic extent, and if you look at the 2019 global assessment of the International Platform for Biodiversity and Ecosystem Services, IPBES, you can see one of their central graphs, which is the one on the amount of species threatened in different degrees in different types of organism groups. And you see that depending on group, this goes from a few percent to almost half of the known species. And you might have seen when it was published the headline that over 1 million of species is potentially threatened by extinction, according to this assessment, which is currently updated. However, this will not stop. It's actually continuing, and it will also continue in the future. And we are already committed to a change that will continue some of these dramatic processes. And I wanted to highlight one paper here, which I'm not involved in, but which comes out of this new Helmholtz Institute. Dorothee Hodap published it last year, where she looked at habitat preference data for more than 30,000 marine species, mostly animal species, and looked at the consequences of different RCPs on the distribution of these species. And what you see here is in the columns, the 3 different RCPs. And then just to highlight a few numbers, in the middle scenario, almost a quarter of all species will actually develop equatorial habitat gaps. So species which have so far had a distribution range on both sides of the equator will have 2 subranges with an intermediate gap. In the more negative RCP 8.5 scenario, almost 50% of species will lose 50% of their habitat. And this is not even the worst scenario, because in this model, which is a classical species distribution model, species codependencies are not included yet. So if you look at the turnover, so the proportion of species that will be exchanged, we go up to 60 or even 80% of species in equatorial and polar regions that will be exchanged. And we see a preponderance of species losses, which are here in the red curve around the equator, whereas we see species gains in high latitude boreal areas. And people working on marine subjects might have heard the term Atlantification of the Arctic Ocean. So species moving poleward. So there is a massive reorganization of biodiversity in all three realms: freshwater, marine, and terrestrial. And one of the conclusions based on this fact has been that we already transgressed a threshold for a safe operating space. And in Rockström's famous 2009 Nature paper on planetary boundaries, you see that biodiversity loss, which is roughly at 7 o'clock on this circular diagram, actually is considered as one of the two things that has exceeded these planetary boundaries the most, next to the nitrogen cycle. The remainder of this talk is about asking, what is such a threshold for biodiversity loss? And is it actually a threshold of biodiversity loss, or is it a thresholds on pressures on biodiversity? And if we use thresholds, what happens if we try to identify targets? Just to start your thinking about it, we can think about two ways of thresholds. One, which is on the top graph here, says that there is a response of biodiversity to some pressure, some stressor. And this biodiversity response is most likely nonlinear. And we could identify a threshold as a pressure strength in which this nonlinear response of biodiversity becomes evident. So our target as a society could be to limit the pressure to a pressure strength that does not incur this massive biodiversity change. Alternatively, you can also think about a pressure of biodiversity change itself. And then on the y-axis, like in the lower diagram, you have some ecosystem property or some ecosystem process, some functionality that you would like to keep and maintain. And you can ask yourself, how much can biodiversity change without jeopardizing this functionality, without risking that the processes or the properties are not longer intact? Of course, these two things are very much related, because the biodiversity change on the y-axis in the upper diagram is the same as the x-axis on the lower diagram. But I think for really clarifying what we mean with the boundary for biodiversity change, it's good to note that you can place such a boundary on two different things, on an axis of pressure and an axis of biodiversity change itself. This lower diagram has been discussed quite a bit. And the idea would be that we have a safe operating space, as you can see here in green, and we have a critical transition, and the threshold is dividing these two things. And very early research on the relationship between biodiversity and the functionality of ecosystem does even indicate a relationship like this. This is from a now more than a decade-old paper by Brett Cardinale, where he looked at primary producer biodiversity loss studies and looked at how many percentage of these follow any of the three potential pathways, like a proportional loss, which means for every species gone, you lose some proportion of the functionality. An immediate catastrophe, which would mean that if you already start losing biodiversity, functionality will be impaired. Or what is called here the rivet redundancy hypothesis, which allows for some redundancy in how biodiversity is linked to functioning, and you only see a decline in functioning after quite some of the biodiversity has been in decline. And you see that most of the studies done, 79 of those who looked between biodiversity and biomass production, 89 for biodiversity and nutrient uptake, 61 for biodiversity and decomposition rates, they indicate this type of rivet redundancy that in a mirror-like image is also in my little diagram here. However, this has been questioned quite a bit in kind of a second wave of biodiversity ecosystem functioning experiments, because it became quite quickly clear that looking at a single ecosystem function at a time is underestimating the importance of biodiversity. Biodiversity is not only driving one function, it drives all the functions that have a biotic component in ecosystems. And if you actually look at more than one function, this rivet redundancy concept disappears and is actually replaced by a much more linear relationship between multiple functions and biodiversity. There are many papers out of this. There are a few really good ones, also by Forrest Isbell, for example. I chose one here by a colleague from Munich, Sebastian Meyer. He looked at the Jena biodiversity experiment manipulation. And Jena is special because there are so many different functions measured. It's a long-running manipulation of biodiversity. And it turns out the more functions you take into account, the more linear the relationship between biodiversity loss and the loss of multifunctionality becomes. So instead of having this safe operating space clearly defined by some redundancy, real ecosystems are very much characterized by a much more gradual relationship where immediate responses to biodiversity loss can be measured. However, and this talk has a few science philosophy aspects in it as well, even if there would be such a safe operating space, who tells which of this biodiversity we can lose? For example, I am extremely fascinated by Central European and Northern European orchids, and I simply don't want them to go. There is an aspect of biodiversity for itself here, which is completely countercurrent against this idea that we have a biodiversity loss that we can tolerate because this biodiversity is redundant. So I'm quite critical about using a threshold for biodiversity loss itself. And later versions of the planetary boundary concept actually have already responded to similar concerns and have changed this in not creating safe operating spaces for biodiversity loss, but for diversity itself. So with that, I would like to move to the other concept, which perhaps is more straightforward in thinking, and that is to limit pressure to a strength that is below a threshold such that biodiversity can cope with this threshold. And when we look at such data, the best examples we find come often from foundation type of ecosystems, where single species create the habitat for the other species. So foundation species systems are, for example, seagrass meadows. Without the seagrass, there's no meadow, and there's not the associated fauna and flora. Kelp forests would be another example, for example. So here we actually see the transition from one type of biodiversity to a different type of biodiversity. And that might be the case where a limit on eutrophication, overfishing, or disturbance can be identified that prevents this slip into a new state. As a plankton ecologist, however, I should make this point, perhaps at this point, that of course this assessment of change from a state that we want to a state that we don't want is a biased way. Because all the dots here that appear on the right-hand side, which potentially Emmett Duffy meant to be plankton, that is, of course, a lot of different species, and might actually harbor very strong phylogenetic and taxonomic diversity. So a second philosophical aspect is it's extremely hard to discuss about biodiversity change without values, without biased evaluation of different types of species. But my main point here is that these type of foundation systems are actually special. And if we look at other time series, this is just one example I've been involved in, led by Gavin Rishworth, where we looked at time series data from Germany and South Africa, coastal monitoring time series for 4 different groups of organisms. And what we see in systems that are highly anthropogenically overformed, we see actually mostly gradual change in biodiversity. We don't see this sudden flip into something else. So what you see here on the x-axis is the temporal distance between 2 time points in the same monitoring series at the same site. Each of these comparisons looks at how much proportion of the species are the same at these 2 time points and how much has been exchanged. So on the y-axis, 0 means exactly the same composition is present at both time points. A 1 means not a single of the species from the first time point is still there in the second time point. And you see that in most of these time series, you see this gradual increase in this turnover. So the farther to data points are away in time, the more distant is their relationship. But as I said, except for very few, you will not see this very rapid transgression type of change. And we have done this now for quite many data sets in number of different projects, and they all rather indicate gradual change in these types of communities that are not foundation ecosystems, rather than critical transitions. But even experimentally, if you really change conditions dramatically, what you mainly see is a gradual change. So this is another study by Dorothee Hodap. It's coming from a globally replicated grassland experiments, the Nutrient Network. It's 50 sites worldwide, exactly having the same type of manipulations. So we have a control, we have a fence that excludes herbivores, larger herbivores. We have a fertilization treatment, and we have the combination of the two. And again, what you see on the y-axis to the left is turnover. So 0 again means no change. 1 means complete exchange of the community. On the x-axis, you have the years after the treatments began. So we are looking at the first 5 years of this experiment. And we are seeing that the turnover gradually increases for these 4 different treatments over time. And that is in response to some of the most discussed drivers of grassland biodiversity, which is nutrient availability and herbivore presence. So it seems that even if we suddenly and abruptly change the environmental conditions, we are not seeing the same type of abrupt change in biodiversity. I recently teamed up with colleagues at the University of Bremen, where they have a really cool paleoecology department. And Tonke Strack, who's first author of this paper, they came up with this idea to analyze the community response to the last deglaciation. So what you see on the x-axis in panel C is the temperature before presence in 1,000 years, and the Atlantic temperature anomaly for this time period. So we see that roughly 17,000 years before now, we find the beginning of the deglaciation. Then roughly 12,000 years before now, we define it as the beginning of the Holocene. And roughly 9,000 years before now, we have a stabilization of the temperature. Then we looked at the composition of foraminiferans, unicellular zooplankton that has the nice tendency to sink after dying and leaving a microfossil record. So we have quite some cores from the Atlantic Ocean, as you can see in panel B. And in panel A, you see the species composition in response to time. Each of the gray lines is one of the cores, and the black line is the assemble average. And what you observe is that with the onset of warming, there is a starting change in the composition, but it actually doesn't stop when the temperature equilibrates. It goes on for quite a while. It becomes even more visible if you look at panel D, where the temperature anomaly compared to present days and the compositional data as a PCA score are directly correlated to each other. So half of what we see as compositional shift actually appears over the last 10,000 years, even so the temperature doesn't change very much anymore. So this highlights that the pressure in inducing some biodiversity change and the response of biodiversity might not be synchronized very well. So when we have this continued change, even though the pressure has already stopped to change, this leaves the question: where would we see the threshold? Where would we put it if there was a nonlinear response? And this leads to the issue of inertia and delayed responses, which I think has been highly overlooked in the debate on biodiversity tipping points. And this is mainly work by Lucy Koscinski, postdoc in my lab. She teamed up with Vicente Ontiveros from Barcelona, a modeler. And this paper has shifted my view on how to deal with this biodiversity topic quite a bit. So the thought starts by looking at different scenarios of environmental change. In the A scenario, environmental suitability decreases for all species. So we do something bad, like putting a toxin into an ecosystem. And we have different number of species that were present before, and all of them do worse afterwards. But it takes time to record extinctions. And that is a phenomenon we know from literature. Tillman in the 90s published this famous extinction depth paper. So we have not immediate extinctions, but we have delayed extinctions based on the negative change. The same is actually true if you do something good to the environment, like protect it or improve conditions. And it's actually something that is really important to discuss in a management context. I've been involved in a number of projects where the success of measures needed to be evaluated. And the project ran for 3 years with organisms that have annual life cycles. We also have an immigration credit. We have delayed colonizations because species have actually to reach this new restored habitat. They have to be able to colonize an improved environment. But the really fascinating thoughts start if you think about something where the environmental suitability changes so that some species profit and others are negatively affected. This means that the relative timing of colonization and extinction is very important to evaluate how biodiversity is changing in this system. Our impression, our gut feeling tells us that if there are as many winners as losers, then biodiversity should be constant. And Lucy's work kind of made me realize that this gut feeling is wrong. So she came to me with an analysis of European freshwater fish data. We used the really great Reef Fish Time database that Lisa Comte and others put together. And she simply looked at species richness over time here. And each of the gray lines is one of the 3,000 sites in Europe. And the blue line is the average, and the blue line is increasing. So the data show an increase in the number of species in freshwater fish in rivers in Europe. Whereas you can read everywhere that freshwater systems and long-lived organisms within freshwater systems are the most negatively affected by global change. So how can that be? First thing we did is we did a classical null model. So we randomized the occurrence of time points and compared our slope against the slope that we would expect from this randomization. And indeed, the randomized slope is 0. A classical null model would predict exactly this constancy of diversity over time. But then we realized that we actually have a very strong autocorrelation here. A species that has a certain abundance will not be gone the next time you look at this system and then reappear with this high abundance again. So we have to induce some degree of autocorrelation in our null expectations here. And Lucy and Vicente did that. And to our big surprise is we got all kinds of dynamics. These are 3,000 runs of a meta-community model. The meta-community model has no driver, just has random drift in the species. So there's no expectation that there should be, on average, a directional change in the number of species. But there is. There is one which is even highly significant. So we see, on average, that in such monitoring situations, because colonization is faster than extinction, we tend to see a positive trend in species richness. And Lucy came up with a measure which we called NICE because we found it nice. NICE is the acronym for net imbalance between colonization and extinction. It can go from +1 to -1. Plus numbers means that the that colonization is faster than extinction. And we see that it's actually positive all the time, both in the data, which is on the left-hand side, but actually also in the neutral model that shouldn't have any bias. And the reason is, as I said, when we start monitoring, we have actually species, and it takes time until they go extinct. And all the other species, before they go extinct, they need to have colonized. So colonization is, as a process, coming before extinction. And that means that our baseline to compare the response of biodiversity to pressures to is not 0. It is actually some positive slope. And we are currently working on ways to predict the size of this bias to help removing this bias from time series analysis. You could say, of course, this could be a very transient process. It could just go away after a couple of years when extinctions catch up. So we actually tested this for both fish and also for bird data, and looked at how long does it take until the bias towards positive trends goes away. And it turns out that for fish, it takes up to 4 decades, and our average monitoring time series length was below 15 years. And also for birds, it takes a decade or even longer in a neutral model. So with short time series, we are actually looking at very short responses of biodiversity and much too optimistic evaluations of biodiversity trends compared to reality. So what seems to be a very straightforward idea to say we have to keep pressures low enough to not allow for critical transitions in biodiversity, in reality, obviously is not. First of all, we don't know how many of these transitions we actually would observe, because it seems that gradual responses are much more common. The trigger where these transitions are initiated might actually be at much lower and earlier pressure strength than the observed biodiversity change would indicate, so that we would have an extremely high security buffer around this estimated pressure strength. But for the last point of my talk, let's quickly assume that there is actually such a threshold. And then the big question is, do we actually, are we able to predict it? And with a team of co-authors, we try to do so using meta-analysis. There's a very nice comment on our paper, critical comment by Steve Lade from Resilience Center, and we reply to that just in case you are interested in this topic. I think it's also worth reading these two. Our idea was to say, in many time series, the reason why we might not see critical transitions might be that the pressure simply hasn't been high enough yet. But that doesn't help us in management. Showing afterwards that there was a transition is very bad. We want to predict it. So our idea was to look at experimental data, because there we have future conditions. So each experiment looks at some pressure, for example, warming, and compares a control to a warm treatment, and we can have an effect size of this warming on the response that interests us. And if everything is gradual, we would assume that with increasing pressure strength, we would also see a higher response magnitude. However, if we transgress thresholds, at some pressure strength, we should see new types of responses. So very low pressure means all systems, even the sensitive ones, are below thresholds, and we see some type of response. But if we have a very strong pressure, we actually have transgressed thresholds even in the most robust systems, whereas in the middle we see a mix of different responses. So we looked at 36 global change meta-analyses. Each of these meta-analyses had one certain pressure, like warming or something else, and one certain response magnitude. So Y and X axis were always the same. But between the 36, we had kind of all major pressures you can think of. And we had 3 different tests. One simply looked for a relationship between response magnitude and pressure strength, independent whether it was a threshold or gradual. One looked at this increase in variance, and one, coming from regime shift literature, looked at multimodal response magnitudes. Most of the meta-analysis actually showed a relationship between pressure strength and response magnitude, as we would expect. Very few, 8 out of 36, showed an altered variance with pressure strength, and not a single one showed a multimodal distribution of responses. From the 8 in the middle group, actually 5 showed a pattern that was not constituent with the hypothesis. 2 actually showed a decrease in variance rather than an increase, and 3 showed an increase only at the highest very artificial pressure levels, which is kind of trivial. You can, of course, hammer each system so much that it flips into a different stage. So actually only 3 out of 36 showed threshold transgressions. Bex Bay wrote a paper a year later and speculated that one of our issues might be that we had log transformed response magnitudes. However, we already had done these simulations in the paper, so that was definitely not the cause. I would like to finish with saying that this is not to promote another locked-in debate where people have different stances and different perspectives and say there are thresholds, there are no thresholds. I just wanted to initiate a critical debate about whether this concept of critical transitions of biodiversity is actually useful in our discipline, and whether it actually allows us to come to better management for biodiversity change. So thresholds are easily communicated. However, I find them questionable for biodiversity change because they are hard to predict. There's a lot of inertia and a lot of gradual change that we underestimate by thinking about static thresholds. And the fact that I'm not able to offer you an easy alternative, what could be done better, doesn't actually make the question less relevant. By contrast, it's so tempting to think about this, allowing this amount of change, this amount of pressure, that it's really difficult to stop this type of thinking and think about this topic in a more holistic way. I put up a few ideas for things that should be done next. And I would like to end with thanking all my co-authors, the funding agencies, and of course, HFMP and Plankton Ecology Lab people. And happy to take your questions.
Thanks very much, Helmut. Virtual round of applause from everyone, I'm sure. Yeah, there we are. We have to keep questions a little bit short today, but we do have a couple of minutes for questions. And I see we already got one from Anita, which is a bit long. Do you maybe want to come on the microphone, Anita, and ask Helmut yourself?
Yes, thank you. Hi, Helmut. Thank you very much for your talk. It was really great. And I was just wondering on the use of the term threshold. It's probably is my perspective, because I would normally associate the term threshold to a management context. So it's where we decide there is a one or more cutoff level where that distinguish the acceptable and non-acceptable conditions for us. And this acceptability can be for anything, can be for, for functioning of the ecosystem, can be for our ability to gain goods and service and benefit from it, etc. So I was wondering whether, and I don't know if my actually in the last part of the talk you were talking about a critical transition. So is that correct to assume that critical transition is a synonym of threshold?
No, for me, the transition is on the y-axis and the threshold is on the x-axis. So however you define it, you set something that is kind of the cause of something, and you set a threshold for this cause. And what you observe in response to this cause is either a gradual change or a critical transition, or if you want to use that word, a tipping point. I quickly read your question, and I think, yes, it becomes super relevant, especially in a management context. Threshold. So the whole thinking about this was actually started by a management project where we were looking at eutrophication thresholds. And I was hired in that project to look at, are these thresholds set at a level where we maintain biodiversity? And it turns out that we can't say that, simply because of all the uncertainties I described, but also the type of response. Biodiversity is never constant; it always changes. So what is the critical transition here? So there is this temptedness to define clear values, 2.8 milligram nitrogen per liter river effluent. But where does that number come from? And does it something good or does it something harmful? Is kind of the question I'm throwing out. And as I said, I'm not in a situation yet to come up with a better alternative, but I see, especially in the management context, a strong bias of defining these thresholds and then everything is fine. And I get worried that there is not everything fine after coming to these thresholds, because we might set them in the wrong places. I hope that was clarification enough.
Yes, thank you very much. Yes.
Thanks. We have a minute for a quick question, hopefully, from Mike before Helmut has to run to his lecture. So.
Okay, thanks, Stephanie, and thanks, Helmut. That was a great talk. Yeah, you mentioned there about the management aspects. There's one of the Horizon Europe projects that we're involved in at the moment, Guest for Seas, where we're looking at these thresholds. But the quick question is, you get the examples from terrestrial and freshwater areas and from the open sea, the marine. What I think many of us also try and get our heads around is these ideas within estuaries, where the organisms are adapted to these highly variable systems. And I wonder if that's what you meant by inertia, that in an estuary where the organisms are coping with this, what we've called in the past this stress subsidy continuum, they're coping with those high variable conditions. Does that mean we're dealing with longer timescales in inertia rather than shorter ones?
I think that is a brilliant question, and it's actually something we start to look at as well. By the way, I'm in your neighbor Horizon Consortium Act now.
Oh yeah, that's good. That's good.
So we are tasked with very similar questions, and out of gut feeling, I would say that species that have experience or are evolved towards experiencing highly variable conditions, they will generally have a longer time span before they go extinct, because the change in mean is actually slow compared to the variability they are observing. And very at the beginning, I showed this paper by Doro Hodap, where she looks at the gains and losses based on a species distribution model. And the losses are especially tropical. And the main reason is that there is, of course, not even a warmer condition where species can invade from, but it's also that the upper limits, for example, of temperature change, the species are so much closer to their optimums, so that any increase will definitely result in a negative growth rate response. There's a beautiful paper from 2019, I think, by Malin Pinsky in Nature, where he very elegantly shows that marine species actually suffer more from projected climate change because they are on average living closer to the to their temperature maximum and optimum. Just for those who would like to read more, there's one paper that I led where we put some of these thoughts into a paper form. So if you want to read them. And there's a new one led by Len Fisher about some of the targeting management threshold ideas. So that goes also a little bit back to Anita's question. And I would be able to take one or two more. So
okay.
Sorry, I took a little bit over time. So
I think Heidi had raised her hand. It's gone now. Yes. Heidi, would you like to ask a question?
Yes, sorry. My computer's doing dodgy things. Thanks for a lovely talk. I must admit some of it did go over my head. My question is, how do you identify the stressors that you're working with? I did miss the first part of your presentation, so I'm sorry if you mentioned it then.
No, at this level, it was very general and very unspecific. We are in most of what we did so far, we are looking at single stressors or low number combinations of stressors, going perhaps up to 3. But Anita already mentioned that in her question as well. There is, of course, additional uncertainty around the cumulative impacts of multiple stressors that might even push this this additional security line that we might need even further to the left, right? Because we are not, or we don't know so much about some of these cumulative impacts yet. But it was not about a certain stressor. We are mainly looking at eutrophication, warming, overexploitation. Recently, a new project about sediment management in coastal areas, which is also a major issue, at least here in our coast. These are examples of what we are dealing with.
Brilliant, thanks. It sort of ties in with what we're doing as well, so it's really interesting. Thank you.
All right. Thanks very much then again, Helmut, for this super interesting talk. I agree, it's really important to have a look at this sort of big picture concepts every now and then, whether you're from a theoretical ecology background or in a management position. So yeah, thanks very much. We'll let you run to your lecture now. For everyone else, I just wanted to quickly highlight next month's talk. Will be on, let me check this, 6th of June. Lucy McMahon from Manchester Metropolitan University will talk about carbon sequestration, so maximizing blue carbon through salt marsh restoration. So a really interesting topic as well. And seeing Tim, I think maybe we should also highlight that it's still possible to register for the ECSA conference and that we have a travel grant. Do you maybe want to say something about that, or should I?
I will say bye at this point. Thanks for having me. It was a great pleasure. And,
thank you.
Bye.
Bye.
Bye, Helmut. Thank you.
Yeah, maybe just a few words. Deadline for submission of abstracts is over, but we still keep it open for submission of poster abstracts because we have to start creating a program, but everybody who wants just to come and present a poster can also submit his abstract still for some time. And yeah, now we're just in the process of reviewing abstracts, and then soon accepted authors will be notified. I think next week, sometime next week, I guess so.
And the travel grant is available for ECSA members, right? So there's more information on the ECSA homepage, I guess.
You find it on the homepage. There's also a hint on the conference homepage that leads you to the ECSA homepage. Yeah, but there's travel grants available, and I hope to see many of you there in Hangzhou.
Great. Then, yeah, thanks for that. Thanks everyone for joining, and I hope to see you next month then for Lucy's talk. Bye, everyone.
Thanks a lot. Bye.